So I would like to introduce you to Mark Iradukunda over here that is ready to deliver his exit seminar for everyone. So Mark has a degree in integrated science from University of Nebraska-Lincoln, and he obtained his degree in 2022. So Mark had been working with a very exciting system. So he has a lot of ground to cover because he did a lot during his master's over here. So Mark originally started with Mark 1 year or so, and I had the privilege to work with him in the last year and saw his development, his growth, and everything that he accomplished. So, Mark, without further ado, I would like you to really highlight what you've done here. And I'm pretty sure that Mark is, listening to you somewhere, wherever he is right now. I imagine here, right, Lynn? Yeah, so.
See,
Uh,
yeah.
All right,
Wonderful.
so go for it.
All right. Thanks, Ronito, for the introductions. Usually when I start my presentation, I like to compare seeds and plants, with eggs and chicken, which came first. Well, I don't have an answer yet, but usually I say, whichever started, one begets the other. So if you get a good seed, you got a good plant. So we worked on image analysis, and we wanted to develop an imaging tool that can quantify the seedling vigor and germination. And to get started, I will start with a few definitions first. So seed vigor, so we're looking at the seed quality, and it's to be stored, how you, when you have a good seed, you can trust that you can store it for a long time. And when the time comes to plant it, you can trust it will germinate well. And that is how you can understand the seed vigor. And anyone who stores seeds understands that. There are a lot of standardized tests, but those are slow and expensive. And of course, you have, to do a seed test, an acceptable seed test is tough. You have to be approved. But for many researchers and growers who don't have those kind of things, we're trying to develop a tool that they can quickly use to get that information easily. So, then to move from a seed, you have to germinate it, and we'll define germination as the resuming of the metabolic activity, so the breaking of dormancy, and that will be influenced by the seed itself, but also by the environment that surrounds it. And that is very important since we are in a controlled environment. All right, so have a seed, we germinate it, now we have a seedling. And for it to be called a seedling first, it has to be able to photosynthesize. All right. So, we want a big seed with a big size, and we look at the growth rate of the seedlings. And all right, so normally with growers, when you start your seedlings before you transplant, you choose the best looking plant, and that's the assumption that the bigger seedling will give you a good plant, and that is very intuitive. So all we're doing really is adding a number to that. So there's nothing new inventing. So the assumptions that growers already have and the technologies that already exist, but we're combining all of that so that small researchers and growers can be able to evaluate their seed lots. And we like imaging because it's quick, it's very cost effective, and there's a lot of growing potential because as you can see, technology every day, something new comes in, and many people are adding new features. So we want to use that advantage. And I want to talk about our program, the logic behind it, because I will talk about image analysis hundreds of times, but I want for us to first understand a little bit of how it works. So first of all, you have an image, whatever kind of an image. And here I'm using an example of a Raspberry Pi camera image. So it's a normal colored image. You feed into the program, and we're using Python, which is open, so free software. That's as a plus. And what that does, it looks at that image, it's a grayscale image, and then it will pick up the pixel intensities, and we separate the background from the plant by thresholding. So we look at the pixel intensity that belongs to a plant. So that's why we have the red line. It's just we want to select the pixel that belongs to a plant and then ignore the rest. And what that does, it gives us a mask. We only see the plant material, and we only see those pixels that belong to a plant. And we can use that to do some mathematical calculations to get the canopy size, to count the plants, and all other calculations that, if you know a lot of math. And in the case of Raspberry Pis, you can, the process is repeated, and you can get your data on your cloud on whatever tool you're using. And then important variables. And I'm studying this early because all my studies are very similar, and we collect the exact same data. And so the first thing, of course, we count the germination. So, and then that's how we get the germination percentage. Then we collect the canopy size, and then throughout the time we do multiple harvests to be able to validate, to correlate the canopy size and the dry weight. And of course, at the end, we have to do some statistical analysis to, show that something happened. And if you like some mathematics, we have some equations. And so the first one is to transform pixels into centimeters squared, something that we can all understand. And then there's also a logistical regression model that we used in predicting our dry weight from canopy size. And this itself was a long process, but and then the growing conditions very similar in our vertical, vacuum growth chamber at 25 degrees C, and lighting was 250 ppfd for 16-hour photo period, and irrigation was automated, and all the seeds were from the same vendor, John Seeds, and we had to select a new and all green, and we will see why later. All right. To develop this tool, the first step was to understand image analysis. So we explored the system using choropleth fluorescence imaging, and then after that, we applied the knowledge into Raspberry Pis that could be integrated in the growing conditions. And then to validate that, we had to do multiple seed tests that are standard, and then look at the results if our tool can pick those variations. So, and then that's where we go to our first study. This one was a little longer study, and to get started, Jonathan and Kellen, especially at the beginning, as I said, I didn't know anything about coding. So I had to learn a lot, and a lot of Googling with ChatGPT. But the objective of the first study was first to understand this non-destructive and automated imaging of the seedlings. And so to do that, we have to measure the canopy size, to quantify the seed germination, seedling vigor, and then to use the canopy size to predict yield. And then I integrate that knowledge into Raspberry Pis. All right, so the first trial using choropleth fluorescence imaging. Our workflows are very simple and similar. So we selected 9 lettuce cultivars, all green, and 4 applications. So we took images 3 times a week, and then at day 14 we transplanted those plants. So half of them we transplanted, and half of them we collected the dry weight, and then at day 40 we did the final harvest. All right. So the first system is a custom-made tent we have in our growth chamber right there, and so it is equipped with LED lights. So you get your plants, you put them in, you close the tent, you provide a blue LED light to excite the plant, trigger the photosynthesis, and then the plants will emit light. And we have a camera that has a long pass filter that will only see the fluorescence from plant material. And that is actually really neat because it does the first step for you. So it already separates the background from a plant material. So this is what we see, and that's the tent, and this is what the first image that's what it looks like even before any further analysis. And that's really nice. And of course, you download them, and then we use our Python codes to generate the histogram that helps us segment for plant and a lot of numbers. And the most important numbers for us are the germination percentage and the count and the canopy size. And of course, we use that information to make graphs. So the first thing we look at is the germination. And I will just emphasize again, our tool is has a very nice feature in a way that you can do those multiple tests on the same plants over time. So the first one is the germination test. And it just looks at the germination over time, and you can compare different cultivars. And just real quick, we can look at the little gem and Elema, those performed a little poorly than compared to other cultivars, while the mule had much higher, much faster germination. But they were all able to reach the desired germination. And we said 90% is a good germination for lettuce. And this number is changeable, so you can put to any number you would like. And of course, to the canopy size, we not see a whole lot of significant differences except on day 8. But you can see the differences in the canopy size increase over time. And so the cultivar that was doing very well in germination continued, of course, to show some improved canopy size. Oh, this thing doesn't work on me. All right, but you can see that cultivar mule at canopy was also doing better at germination, and the slow growing cultivar also had a bad germination, and that is something useful for us. I will explain the model a little later, but throughout the studies, we had to figure out the best model that can help us predict, that can follow the growth patterns of the plants. And the first analysis, we only dealt with seedlings. And you know, you remember the equation, I hope. And here you can see that 68% variation of the dry weight of seedling can be explained by our model, and the root mean square of 0.01. You know, of course, we had dry weight, and we observed a lot of variations at the seedling level and the harvest level. But the thing I want to point out is some interesting differences. So at the seedling stage, we had a cultivar mule performing about 2.6% better than another cultivar called Sparks. But at the end, the results were reversed, where this cultivar Sparks proved to grow a lot, to be a lot better at the end. But 17% better, and that was not good news for us. And you know, it was a question, because the assumption again, a big seedling will give you a big plant. So that's, you know, was the first issue. But another issue was, of course, taking up you know, those little trays to the imaging, take them back. That was not automatic. It took a lot of hours, and we did not collect the final dry weight, the final canopy size, and so that's why we cannot say anything about the differences in the dry weight at the final stage. So we corrected that mistake in our next studies. But the variations, of course, you know, cultivars will have different growth habits. But so we still have the same question: does the best seedling become the best plant? I do not know. So to find out, let's stay tuned in and subscribe to our channel. All right. So the next trial, then we applied, we took all those codes and configured Raspberry Pis. So Raspberry Pis are very tiny computers. Yeah, tiny computers, but they're like this big. And there's that blue thing's a little camera. It of course took a few weeks to get those things working. So those are the Raspberry Pis installed in the growth chamber. So instead of having the separate systems, you are growing your plants as normal, then you install your camera, and you do as you normally do, so no interferences. And so that's your camera and cameras and people there. So that's the view of people, but here is the view of the camera. So the camera is overhead. And another nice feature to our tool, of course, you know, you can train it's a lot of coding, but you can have multiple treatments in the same tray and just train the code to go and segment the pictures and do whatever it does. You can even do a plant-based, like Jonathan's worked on that. We can analyze individual plants. And of course, automation. What we see is, we have real-time data of what is happening in your system. And if there are any differences, you can see them ahead of time, but you can see at the end plants start touching, and that is a problem that we will see later. The workflow is very similar. We saw seeds, but this time we only selected 4 cultivars and did 8 replications, and this is because things could go wrong, and they did a few times, so we didn't want to lose our data. And at the end, we did harvest, but this time 3 times, and we collected the data from the time I sowed seed to the harvest. So I have complete data now. But just a few things. First, this is the real-time data. So the Raspberry Pi will be taking images and analyzing them. It's all automated. You don't have to go take images. It's scheduled through Concap, and you can see the changes in the canopy size. And you can already see when we reach to about 14, 15 days, we start seeing weird things. And so the only way canopy size can decrease in this stage, it's if a plant died and we do not see dead plants. That's because plants started touching and leaving the camera view. And so we start seeing weird data, but you can also determine when is a good time to take images and when is a good time to not. Another nice feature, of course, is the germination. This tool will tell you when you reach your germination, and you will compare it to multiple cultivars or treatments you have. And for example, you have this cultivar buell that reached 90% germination about 4 days, but the other one was like 2 days. And those comparisons are very important when you want to understand what's happening with seed lot, when you want to understand what seed lot has more potential to give you a better yield in the future, what can stand harsh conditions. And of course, we did analysis at the seedling level. So we did multiple comparisons on early days of their growth. And you can see differences in the canopy size increase over time, where again, this cultivar mule seems to be doing very well in terms of canopy size. And again, in germination, so it reaches, these 2 cultivars, buell and mule, seem to reach the 90% germination that we want. And for that reason, we selected them for the next studies we did. These other 2 cultivars, the Cristobal and the Crispino, were a little tricky. They not reached the desired germination, but it was decent. All right, so now remember the question: a seedling and the final plant. So at the seedling stage, we can see that this yellow orange cultivar and the red, so the mule and the Crispino will do very well in this little window here. That's the seedling stage. But since we have the complete data, we follow the plant canopy size over time. And at the day 21, they start flipping. So the Crispino and the buell start doing very well, better than the mule, that red cultivar. And at the end, things get much clearer that this cultivar Crispino, in the beginning, grows a little slow, but in the end, it flies away. And it's very easy to see because of the structure of this cultivar on the right. It has very fluffy leaves that fly everywhere. So at the beginning, it's hard for the camera to pick up the complete canopy. And remember, we're taking a 2D image. So we have a better canopy when the plant grows more outward than if it's vertical. So at the beginning, this cultivar is very vertical, and then we see less canopy. But in the end, the leaves start falling, then we have more canopy. And so that's why in the end, it had a much better canopy. And actually, this was reflected in the dry weight. Crispino had a much higher dry weight. And we continue to see significant differences among us cultivars in dry weight, as we saw in the canopy size. And so, you know, as the plants grow, the variations increases. All right. Well, the big thing is, you know, we can actually predict dry weight from the canopy size, and about 88% of variations can be explained by our model. And a lot of variations at the end are due to the cultivar. And then you would wonder, why didn't we just analyze for each cultivar and for each day? Remember, the goal is to have a tool that can work in your system, and you might not understand a whole lot about coding or all this modeling. So we're trying to find the simplicity, the most simple tool, the most simple model that you can use in your system, and get decent results. And 0.88 R squared is very decent, and actually it will keep increasing. And yeah, so you can have a lot of information from this, so you can know your growth rate over time. And this is a complete model from seedling stage to the final harvest, and when you reach your maximum growth rate. So yes, we were able to get to monitor seedling growth, and then but we saw some inconsistencies in dry weight and canopy size, and then a generalized model is good enough. All right, so then we start incorporating some fun things. Seed aging, just like any biological thing, seeds lose quality over time, and that is influenced by its own metabolic activity, but also the storage conditions. And we can explore that instead of waiting for 10 years for the seed to lose quality, we can accelerate that aging by increasing the temperature and the humidity. And this is already existing standard seed test, but it's more appropriate on bigger seeds like corn and soybean. So there is this saturated salt accelerated aging, which is more adapted to smaller seeds. And that is because of their small size, surface, they can absorb a lot of moisture, and you will have problems. So instead of 100% humidity, we have the 76 relative humidity. So yeah, we want to see the impact of that aging to the seed and seedling vigor. And then, of course, we hypothesize that aged seeds will show diminished germination and canopy size, and the more time they spend in those horrible conditions, the worse they get. And the workflow is sound. So we did seed treatment, and this time it's only 2 cultivars, sowed our seeds, image analysis, as you saw, very automatic in the growth chamber, and then pre-harvests. Very simple, just like the previous study. So to age the seeds, well, we made a solution of 40 grams of sodium chloride in 100 ml distilled water. And so this is not soaking seeds in the salt. That is a different test. This we just, you have a germination box, and you have a solution in, and then you have a wire mesh at the top, and you have your seeds there. So, and then you close it. So it's just to contain that solution, so that with increased temperature, because we had like 41 degrees C, which is a very high temperature, and that increases humidity, and it's trapped in, so the seeds are quite stressed. And what did we see? So on this cultivar wall, when the seeds were exposed for longer periods of time, so that red line, 72 hours, they did really horribly. They did not germinate well. Though all of their treatments could not reach 90% germination. But on this cultivar mule, it's slightly different. The negative impact of aging started happening at 48 hours and later, but 24 hours did not really become a problem. So it's very cultivar different. And how about the canopy size? So we see the control, the salt, and the stress over time. And we don't see much of a differences, except when we reach 72 hours of the stress. That's when we see very significant reduction in the canopy size. And the surprise here is that exposing seeds for 48 hours produced better plants, and that is very surprising. And Yeah, I haven't figured out why, but it might be with the priming. So I have to look at, be able to separate the stressing and then the priming. There must be a very fine line there. But it's very clear that when we exceed that priming line, the seeds start dying. In fact, like on this cultivar, they really did reach maturity, and they did not actually germinate, and I will show you. And so Canopy says when we follow it, we continue to see that on that red line that they really were suffering, but still the 48 hours did well, 24 hours did well. And we didn't see much of differences at the cultivar mule, and from the literature, so even the vendor, when they sell heat resistant cultivars, mule is one of them, and we saw that in the literature. So it's very resistant to stress, so it makes sense why I would not care too much. Maybe I should have increased the exposure time to the cultivar. But the important thing, and I like here, is the consistency. So a treatment that was big at the seedling level was also big at the harvest level. Which when we come to that question, that is a big seedling, will a big seedling become a big plant? If you are in that cultivar, that is yes, in this study. And we will continue to confirm that from the rest of the presentation. And so when we look at the dry weight, we did not see significant differences in the dry weight in this cultivar mule. But we did not see them also in buell, but it's because the plants the seeds that were exposed to 70 hours treatment died. Well, they did not germinate, so the seeds died. So we didn't have data at all. So we excluded that into the further analysis. So yeah, it's very clear that the stress at 72 hours is very detrimental to the seeds, and you should not do that, especially on that cultivar. And of course, our same model again, very good, and we keep getting better. So now we have 93% variation explained by the model, and 0.15 remain square. Yeah, but you can already see that those extreme values are from the seed that was just at 72 hours. All right, yeah, so it's very visible that you should not kill these seeds. All right, so now we can conclude that the aging does negatively affect your seeds, but in different ways in different cultivars. And we did not see really significant differences in the dry weight. Well, they died, so we couldn't really do anything. But a bigger seedling gives you a bigger plant. And that is very important. That's why we're smiling. All right, last study. All right, this one, so the last study was to stress the seeds. Now this one, we want to stress them, but we also want to unstress them all at the same time. So what we did is we applied salinity, and this is a very common test. Oh, another thing is all these tests were done at the seed level. We stress the seed, then we plant it. We don't do anything later. So, salt, of course, the sodium chloride interferes with the membrane and toxicity and ion leakage, and all those are negative things for the seed, so it won't germinate well. But gibberellic acid is well known to help seed germination and break dormancy and help the cell division, and its application in the stressed seeds has been known to alleviate different kinds of stresses. And the objective, of course, is to quantify the impact of those solutions. And so we soak the seeds in sodium chloride, and we hypothesize that we reduce the seed quality, and then applying this J solution will improve the germination. And the more J we apply, the better. Also, granular sodium chloride, very standard, and then J powder at 90%. Okay, all right. So first, we selected 2 cultivars, and then we have the controlled distilled water, then in salt, And then we had this same exact stress, but added a little bit of GA. And then lastly, same source salinity stress, then a lot more GA, so 150 milligrams per liter GA, and then 50 milligrams per liter. And we store those seeds in a dark for 24 hours at 25 degrees C. And we store them in a growth chamber to monitor those conditions. And shout out to Andres and Andis, and they helped me come up with this thing. It was rough to come up with the right treatments. They seem to be different at each experiment. So it's workflow very same: treatment, sowing, analyzing, harvesting, and analyzing again. And all right, on terminations, we don't see a whole lot of differences, especially on the cultivar mule. Again, this cultivar seems to resist any kind of changes. But on the mule, we still reach the germination, but you can see there's a little bit of reduced germination when we applied salt alone. And on the canopy, again, so this is the control. We apply salt, so you can see a little bit of reduced in size. Then the rest, more or less, nothing very significant. And we did all the tests we could, no significant differences. The only thing is, again, that will answer our question, a big seedling, a big plant, is that, those lines, the line that was at the top on 14 days was also at the top on the last day. And you see a whole lot of variations at the very end. And that's because, it's hard to project the canopy size when there are too many leaves touching, and but still, this is very good news for us. Again, the model, and we improved our R-square to 0.96. So yeah, maybe the next experiment will reach 150%. But yeah, we'll keep getting better and better. And actually, so, that is very easy to see with the variations you induce. And so in the last experiment, we induced the negative variation, but then we kind of negativity. We also, it's like negative and then negative to the negative, we just cancel out, then we were good. So it's as if we didn't do a whole lot of things in the end. And yeah, all this is very standard. So dry weight, we didn't see much of differences except on this cultivar mule at the very end. And actually, surprisingly, GA was bad. When we applied a lot more GA, 150 ppm gibberellic acid, we saw a reduced dry weight. And the interesting part is, and it's not hard to explain. So the control is that on top right, we have a normal plant, then we apply salt, a little bit of decrease, but nothing. Then you have 50 grams, 50 ppm of GA. So you see that the seedling is a little bit elevated, but then with the too much gibberellic acid, we have a lot of hypovertical extension. We have a lot of pale leaves. So even though the plant looks big, this is not a healthy plant. If you were transplanting, especially in the field, you don't want that. It's not good. You want an erect plant for better exposure of the leaf to photosynthesize. And of course, that is the side view, but the camera view is different. You see, if it's laying on one side, we don't capture as much information as we could. So like this plant is really big, but it's not that big. And it's due to the leaf orientation. All right, conclusion. So salinity is not good for your seeds. GA can elevate stresses, but plants are very sensitive to the hormones. And so there's a very fine line between promoting and killing your own plants, phytohormones. And we didn't really see a whole lot of significant differences in the end. Right, so good news, we successfully developed an imaging tool that is cost effective. What else? Yeah, non-destructive, and that can help you evaluate your seedlings. And the benefits, of course, is the cost. The hardware is very cheap, so a Raspberry Pi is below $100. Other kinds of cameras we've had somewhere into like $3 grand, $5, whatever. So that's different. And then it's simple. Well, the starting point is the hardest part. Especially when you didn't have a background in coding but once it's running, like right now I have experiments running. I haven't looked at them in ages. And another good thing is you only get information you want. There's a lot of structural information, and the next presenter will show you a lot of cool things you could do. But we only just selected the least amount of information for certain useful applications in very small operations. And so it's unique because it's integrated in your system, so you don't have an imaging station and then you know the growing system, and then it's adaptable to multiple applications. We have done some leaf variegation quantification. There are some seed vigor, like you can really adapt it to many things based on your knowledge. And so, but it's be careful. We know the protected canopy size can predict dry weight. A generalized model, all days, all the treatments is good enough. And if you want to see the variations, it's better to do them within the same cultivar than comparing different cultivars because they have very different growth habits, and that could throw you off. Two-dimension images are limited in it's only a few information. If you want more information, maybe more cameras or a lot of things. But for the sake of simplicity, this was good enough. And so with that, I would like to thank the department, our lab, the seed lab in Tifton in designing the seed treatments, and the funding from the department, and of course the lab and the American Horticulture Research Institute and our fertilizer donors, and finally, of course, humans. All this was possible because you are my friends and colleagues and family were there to support me, and here we are. Yeah, and with that, thank you.
Excellent job. Questions for Mark?
I don't have a question, but I just have a comment. I really like the flow of the presentation from the unknown to known and the way you articulated the results. Yeah, good job.
That was tough.
Thanks.
I have a question.
Yes.
So in the first trial one, you had stats to be, your model was able to, what do you think your model was able to kind of construct what Sparks is as a cultivar? So what made you, what do you think was wrong with the other cultivars that the model wasn't able to?
Oh, no, so I chose that because the contrast was just too much. So you have at the beginning a certain cultivar doing very well than others, and another cultivar that it's not showing any differences. And then at the end, this cultivar just is very different from others, like 27% from the second best. And so that's why I was like, what happened? And that's what triggered to do these next studies and think about the cultivars is what is happening there. And so yeah, there's isn't really a big reason, just like I was intriguing to see the contrasts. Yeah.
No, click the button on the mouse.
What?
Click the button on the mouse to turn it back on. There you go. Okay.
All right, any questions on Zoom?
Can I ask a question? This is Jong Hyun Kim. You did a pretty good job, Mark. And I think the optimal environmental condition for germination may vary among cultivars. Have you checked the optimal germination environment for each cultivar?
No, I just I looked like generally for lettuce in general.
So the rates of germination and growth may vary depending on the environmental factors as well, and also the cultivar. So if a similar study is conducted under different conditions, such as the different environmental factors, how can the results can be effectively utilized? What do you think about that?
Yeah, I mean, that's a general thing I observed. It's that every study had their own cultivars, their conditions. Well, so for practical purposes, we generalize lettuce likes this temperature, likes this heat, but cultivars, of course, have the variations. But and I really wish there was a very standardized way, like like a catalog where you have, okay, this cultivar likes this amount of temperature, like very specific to cultivar. But to do that, you would have to read a lot of papers, and each paper, of course, will report the different results. But still, it's still very applicable in a way that, for example, this cultivar mule, we observed that in all the treatments we did, it continued to behave very well. So though initially the cultivar differences are an issue when you're comparing a lot of cultivars, for practical purposes, in the end, if you have limited cultivars with similar close enough behavior, you have a good chance of applying that knowledge. So yeah, it's not a great answer, but for the practical purposes, that's the thing I can think of. It's there is an ideal, but for practical purposes, let us, as long as they have a similar behavior, they would be good. But also the vendors will put information on the kind of temperature, the growth patterns, and also how long it will take. So yeah.
Yeah, so thank you for that answer. But one of the regarding question, because canopy size and dry matter that you showed have pretty good correlation, but they have little differences between the color that I'm not sure. But did you see any specific cultivar specific relationship between the canopy size and dry matter? And can we use that to um optimize that kind of calibration?
Yeah, we did see some cultivar influences, especially the second study. However, again, the goal was to provide the most general tool that you can use in your production. So though, yes, there are variations in cultivars, and especially on the second study, as I showed you, when we treated seeds with 72 hours, some seeds died, and that will impact the model. So to calibrate, of course, it's ideal to analyze individual cultivar, individual days. Like that is the best way to go. But our goal is really something general and good enough. So yes, it's true. It's best to do cultivar wise, but for these purposes, we were like, okay, we accept these variations.
We do have a question on Zoom, so there will be a very quick one. So to what degree do you think that your camera method could be used to elucidate trade-offs between different qualities among cultivars? For example, growth rate versus biomass or other qualities.
What degree? That is a hard one. I'll say good enough. But so when we started comparing 9 cultivars, it was a little difficult. The growth habits. So there will be variations, but it will be hard to say, okay, this cultivar 1 is this much different from A, B, and when I did the final harvest, multiple comparisons, they were sharing a lot of information. So if you're comparing too many comparisons, just in generally multiple comparisons, it's very difficult. But I think it's best with 2 cultivars, they'll be better to compare than if you have too many. So what degree is that? But yeah, as you saw, the variation was 68% with 9 cultivars all days. So it's still decent. So it depends on how your goal is, if it's the accuracy or if it's just generalized. Okay, this is generally better than generally worse than that. These are tough questions.
Thank you very much. Appreciate it. We are right on time.